on with the next speaker. We zoom in into nanoscales, molecules. So Koen Martens, he's from the Laboratory of Biophysics and Nanotechnology. Koen?
Yes, thank you, Remco. I'm here really to talk about single molecule microscopy and how we can apply this very new research field to study both life and food systems. So if we take a look at nature, then you can see that nature is actually one very massive length scales of stuff that's going on. At the complete right-hand side, you see the size of humans that are more or less 1 meter in height. And if you go completely to the left, you go to the DNA, to the individual atoms, which are in the sub-nanometer size ranges. Now, as Thomas already indicated, there is this bottoms-up approach, which tells you that if you know enough about what's going on the left side of the screen, you can use it to build up everything on the right side of the screen. So I'm really interested, especially in the region between roughly 10 nanometers to 10 micrometers, because I'd argue that if you understand everything about this size regime, you can use it to study everything that's to the right of this regime. Now, if you take a look at, for example, these images of an eukaryotic cell, these are really textbook images. It's a very empty cell, and only the few things that we are interested in are highlighted and actually shown inside the cell. But a cell is much more complex. Here you can see a very model system of a bacterial cell. So this is roughly only 1.5 micrometers in length. So this is 1/1,000,000th of a meter. And you can see that it's booming with complexity. In the center, in yellow, it's the DNA. On the outside, it's a phospholipid membrane. And in the middle, there are all these different kinds of proteins that regulate the entire cell. And there are millions of them inside. If we would zoom into a very small piece of the cell even, you can appreciate how dense all these proteins are stacked together. And even though they're stacked this close together, they can still completely function and regulate this whole cell. However, if we want to study this, it's going to be much more complex. Because of course, this is very nicely pseudo-colored and everything. You see everything what's going on. But in real life, everything is black and white. So you don't really see where one protein ends and where the next one begins. So we need to have some way to highlight one individual protein to actually study how this protein moves throughout the cell and finds its targets. What's more, of course, the cell is not just immobilized. Everything is always moving, the cell itself, but also all the proteins are moving. So we actually see something like this, very fuzzy, very motion blurred image. So to study this becomes more and more complex with every difficulty that we have. Now luckily, we can take, let's say, very rapid images. If we take roughly 100 images per second, then this motion blurring is counteracted. We go back to an image like this, where we can study where every protein is at that particular moment. The next step is to actually identify this one protein that I imaged here in purple. So we just attach a light bulb to it. If this is the only thing that emits light, then we really know, okay, here is something that we're interested in. Now, of course, this is not as easy as I make it out here. We need to replace a simple camera by a microscope, and we need to replace this simple light bulb by a fluorescent protein, a single protein that can emit light. Now, if we look at how we do an actual experiment of this, you see that we load single cells on top of a rather complex microscope. And you can see in the bottom left, you can see a single light spot moving around in the cell. And later on, we added these yellow lines to actually see how it is moving around. Now, as you can appreciate, this light spot is fairly big with respect to the cell. It's almost 200 to 300 nanometers in size. This is simply because light itself actually has a size. It's in the visible light range, so it's roughly in the orders of 400 to 800 nanometers. And these light spots are roughly half the size of this. However, if we know that one single protein is underlying this huge spot of light, we can mathematically model this spot of light and say where the individual protein is with a precision of 10 to 20 nanometers. So much better than we can do with normal light. Now, if we've done that, we see something like this. We see a cell, a snapshot, where one single light spot is present, and we can determine the center of it. Now on the right there, I'm going to make what's called a jumping distance histogram. So I'm calculating the distance that this single molecule jumps every frame, and I make a histogram of this. So maybe the first time it jumps this far, we get a value in our histogram. Then it jumps again roughly the same size, then it makes a very small jump and a very big jump. And we can build up this histogram by looking at this single protein over a very long period of time. If we do that enough times, we get a very continuous histogram. Now this is what I'll be mentioning in the next slides. But first, we need to check what we are actually interested in. Our first model study that I'm going to explain is on CRISPR-Cas9, as Vera already introduced. Originally, these are used in the bacteriums as an antiviral agent, so it identifies a virus attacking it and can defend itself against it. But currently it's being much more used as actually a way to edit DNA of both bacteriums, plants, and also humans that we're testing. Now for CRISPR-Cas9 to work, it has a very specific sequence loaded on the protein, shown in yellow here, and it needs to find this complementary sequence somewhere on the DNA, which is underlined to the left here. Now it can do this by simply searching along the DNA and checking every spot if this is a good match or not. But of course, as you can see, this is taking forever. So nature has found something smart, which is that it will check for a dinucleotide sequence, a double G sequence inside the DNA. And what it does, the Cas9 protein only binds on this piece of the DNA, and then quickly hops to the next piece of DNA to check there again, and then quickly hops to the last piece of DNA. And now you can see that it's much faster at finding this complementary sequence and doing the DNA editing over there. Now we are very interested in this system. We are mostly interested in how quickly it moves from one state, free moving around, and being bound on the DNA, and how quickly it does this checking of the DNA. So for that we designed an assay, where in the beginning we only look at these two states, where it is either free in the medium, or free in the cytoplasm, or bound to the DNA. And what we could see there, we would expect that if this is bound to the DNA, it's moving very slowly and makes very small jumps. If it is free in the cytoplasm, it moves very rapidly and makes very big jumps. Next, if this switching between these two states is roughly as fast as we can take images, we see a broadening of our histogram. And with this broadening, we can actually quantify how fast it does the screening of the DNA. So this is very nice as a hypothesis. Our experiments look very similar, which is of course very good. So we've imaged hundreds of cells, up to thousands of cells, and in every of those cells we can image 200 to 500 of these single Cas9 proteins moving around. And with that we could make this jumping distance histogram, and from that jumping distance histogram we could use a computational model to fit the speed at which it's binds the DNA, searches the DNA, and is removed from the DNA. So if we put this in the model that you saw before, we can see that's actually very fast. Checking the DNA and checking if there is this dinucleotide sequence happens roughly 30 times every second, and that's going on continuously. And we found even further that if it is bound to a target site, it can be bound there stably for over a minute before it is released again. So you can see that it's a very dynamic interaction. Now what's next? We realized that this is a very powerful technique to use, so we want to apply it to food stuff as well. So I showed here 3 very nice t-shirts, and other than that they're very sugary and very tasty, they have something else in common, which is that they require a very defined structure. And the structure is actually given by something that's called a hydrogel. And a hydrogel, it's already in the name, it's a gel that consists mostly of water. So what you can see here in the schematic in the bottom right is that this gray area is water, and then the small strands that you see are small sugar strands which contain the water together. In light gray, it's a very coarse structure. And if we add some kind of fluorescent molecule or fluorescent particle in there, then those particles can move around quite freely. They are somewhat hindered by those polymers, but most of the time they can move freely around. There also exist very dense structures, and in there, each proteins, or each polymers in this case, are very much more restricted in their movement. So as an experiment, let's do something like this. We can image much bigger fields of view than depicted in the bottom right. And we could indeed visualize these different mechanics. So we could see some tracks, the top one, which move very rapidly, which is in this coarse network. And we could also see tracks that are very slow moving, which is in this dense network. If we look at all the jumping distances again, we can again create one of those jumping distance histograms, and we could clearly identify these two populations. The slow-moving population in blue and the fast-moving population in red. And we could map that back towards our model system, and there we can see the coarse network in red. Now we can use these actually quantified jump distances to tell a lot about this hydrogel network. We could tell precisely how thick these polymer strands are, and we could precisely determine how big these dense network structures are. So with that, I briefly like to discuss on what's next in these fields. Of course, in life, as I told you, the bacterium is complex as ever, and we can do a lot of different proteins and images there. And in food, we're specifically looking at plant-based meat replacers for the next coming years to study them. And with that, I thank you for your attention.
Questions? So you are looking to a single molecule,
yes,
but the light beam is much broader,
yes,
and so the number of molecules in that light beam will be millions.
No. How we do it, especially in light, this is very difficult. We actually have fluorescent proteins that it's a light bulb, which we normally just switch off. And then we have hundreds of light bulbs that are all switched off. But then, with using very high energy light, with UV light, we can stochastically only switch one of those light bulbs on. And then we know for sure that the light is originating from a single protein, from a single molecule, and from there we can actually identify where it is located.
Thank you for this very interesting presentation. What I was wondering is, of course, you identify the fluorescent tag. Do you have to make any corrections as to find the specific location of the protein that you're actually tagging?
So it's a very interesting question, because this, of course, we're not looking at the protein itself, but we're looking at the fluorescent protein that we fused towards this protein of interest. Now, this distance is very small. It's in the order of 2 to 3 nanometers. So this is actually within the error of the localization position that we can achieve. Nonetheless, we have to account for it, especially in our biological systems, because of course we're technically not studying our protein of interest, but we're studying a kind of different protein that we adapted in the laboratory. So we have to take into account that it's slightly larger, and we can maybe have different interactions that we previously wouldn't have expected.
No more questions? No? Now, Koen, thanks again.